Hello, I am Laura and I will be speaking about REMBI and its relevance to annotating bioimage metadata. In this RDM Byte, I'm going to give you an overview of REMBI. You will learn what REMBI is, how it can help you, and how you should use it. All the images in this RDM Byte are from the REMBI publication cited on the next slide. REMBI stands for Recommended Metadata for Biological Images. These are a set of guidelines with the aim to create metadata standards for diverse imaging communities. These are currently in draft form in order to encourage discussion, but they provide a useful guide for considering what metadata to collect so that your image data can be made fair, findable, accessible, interoperable, and reusable. In the REMBI publication, the authors highlight three potential user groups for bioimage data: research biologists, image scientists, and computer vision researchers. These users will all be interested in bioimage data, but for different reasons, and therefore need different metadata. For example, a research biologist may be interested in the biological sample that was imaged in order to compare it to other samples that they are working with. And imaging scientists may be interested in how the image was acquired so they can improve upon current image acquisition techniques. And a computer vision researcher may be interested in annotated ground truth segmentations that can be obtained from the image so that they can develop faster and more accurate algorithms. All of these require different metadata to be available. REMBI gives you a starting point when facing the daunting task of collecting your metadata. It breaks the metadata into 8 modules seen in this diagram. This can be helpful as you can divide the stages of your experiment into the separate categories and focus on collecting just the metadata for that section. This reduces the task into manageable chunks so it doesn't feel overwhelming. This can also be helpful if you work with collaborators, because you can easily delegate relevant modules to each person, so that multiple people don't collect the same metadata. In other RDM Bytes, we will describe each of these modules with examples to explain them in more detail. If you plan to submit your data to a public repository after publication, collecting the information suggested by REMBI, alongside data acquisition and analysis, should mean that you have all the required details already collated for the submission. You don't have to worry that you may have forgotten some details or have to search through multiple directories to find what you need. However, if you know which repository you will use, check if they have a template or instructions for the metadata. You can then use that instead or alongside REMBI. Finally, annotating your metadata in line with REMBI enables others to reuse your data, adding value to your research. So how should you use REMBI? Take a look through all the modules before you start, or continue watching this series of RDM Bytes to get an idea of what information is covered by each module so you don't capture information more than once. It will also give you an idea of what other researchers need to know from you. If you are a research biologist, you may know all about the metadata required for sample preparation, but you may not be aware of what information a computer vision scientist needs to be able to use your images. Use REMBI as a guide. These are draft proposed guidelines aimed at covering a broad range of bioimage data. So if you think something important isn't covered, add it. If you think something is irrelevant to your data, ignore it. Please see links to additional resources alongside this video, as well as other RDM Bytes in this series.